Hey everyone, how's it going? My name's Akash, and this video is going to be pretty similar to our last video. So in our last video, we took a look at the signaling pathway of our messenger molecule called acetylcholine. In this video, we're going to do the same thing, but we're going to look at the signaling pathway of the messenger molecule called dopamine. So as we established in our first video, the signaling pathway goes production of our messenger, packaging and storage, release of our messenger, production of a response, and then eventually this response gets terminated. So let's look at how dopamine goes through these 5 steps. Let's get into it. So before we get into it, I just want to quickly mention that I would really recommend watching my past videos on packaging, release, and termination mechanisms before watching this video, because as we go through these different mechanisms for dopamine, it would really help to have this background understanding and foundation and really help understand this video that we're about to go through. So I'd recommend going through those ones first, but yeah, with that, let's get started. So before we get into how dopamine is produced, we first have to understand that dopamine belongs to a group or a class of messenger molecules known as catecholamines. So catecholamines, this can basically be split into 2 parts. So if you look at the first word, catechol, that basically means that these messenger molecules are based around this catechol chemical structure that you see over here. So the structure is basically the foundation or the backbone of all your catecholamine messenger molecules. And then the second half, amine, basically means that these messenger molecules have an amine group. So that's, you have a nitrogen group bound to a couple of hydrogens. That's an amine group. So they're based around the catechol structure and they are amines. That defines catecholamines. And dopamine is an example of catecholamines. So there's basically a pathway in which lots of different catecholamines can be produced and turn into other catecholamines. So let's look at this production pathway. Okay, so let's say you have a neuron and outside the neuron you have some blood. Sorry, I probably should have written this in red, but here is your bloodstream. So in your blood, you have a chemical called phenylalanine. Phenylalanine or phenylalanine, whatever you want to call it. But this chemical is in your blood, right? And this chemical is basically the starting point of your entire catecholamine production pathway. So a hydroxylase enzyme can come along. Hydroxylase. And as the name suggests, what hydroxylase enzymes do is they add a hydroxyl or an OH group. So what hydroxylase does is it adds a hydroxyl group to your phenylalanine. So now you have your phenylalanine with your OH group attached. And now with your OH group attached, this phenylalanine it becomes a new type of chemical, a new type of compound. And we call this compound tyrosine. So the addition of your hydroxyl group onto your phenylalanine turns it into tyrosine. And once it's turned into tyrosine, it can be transported via some transporter into your neuron. So now you got tyrosine inside your neuron. So now that your tyrosine is inside your neuron, another hydroxylase enzyme comes along. And we already know what hydroxylase enzymes do. They add hydroxyl groups. So this comes and it adds another hydroxyl group. So now you have tyrosine with a hydroxyl group added, and this changes into a new type of molecule called L-dopa. So basically you started with your phenylalanine in your blood, you added a hydroxyl group which turned it into tyrosine, you pump your tyrosine into your neuron, and then you added another hydroxyl group which has now turned it into L-dopa. So before we go on, one important thing to know is that all 3 of these chemicals, phenylalanine, tyrosine, and L-dopa, they are all what we call amino acids. So what are amino acids? Let's look at the name, amino acid. As the name suggests, amino acids are made of 2 different parts. You have your central carbon, your hydrogen, and then you have an R group, which is basically a variable group. This can be a lot of different things. But the main defining characteristic of an amino acid is that you have an amino side and an acid side. Now, amino side you can probably tell that it sounds like amine. And that's because on the amino side, you have an amine group. So this is your amino side. And on your acid side, you have a carboxylic acid. So COOOH. So you have an amino side with an amine group, and you have an acid side with a carboxyl group. So up to this point, your phenylalanine, tyrosine, and L-dopa, they are all amino acids, meaning they have both of these components, an amine side and a carboxyl side. But remember, we said our messenger molecules are called catecholamines, and that's because they have an amine group. So that means we somehow have to go from an amino acid to just an amine. We have to go from having both a amino group and an acid group to just an amino group. We have to somehow get rid of our carboxylic acid group. And that's exactly what happens to turn L-dopa from an amino acid into a catecholamine. We need to remove the carboxyl group. And this is done by another enzyme called decarboxylase. So just like how hydroxylase is an enzyme that adds hydroxyl groups, now we have this new enzyme called decarboxylase. And what it does is it removes a carboxyl group. So that's C-double-bond-O-OH. It removes this. So as we said, your L-dopa is an amino acid, meaning it has an amino group with the nitrogen and some hydrogens, and it has a carboxyl group with a carbon double bond OOH. So decarboxylase comes along, and it removes this carboxyl group, and now you have only your amine group. And with that, it has now become a catecholamine. Specifically, a catecholamine which we call dopamine. And this is your messenger molecule. This is your catecholamine messenger molecule, dopamine. And that basically covers the first step of our signaling pathway of production. We have successfully produced dopamine by taking phenylalanine, turning it into tyrosine, pumping it into the neuron, turning it into L-dopa, then removing your carboxyl group. So it's just an amine, and now you have dopamine left over. So now that we've produced dopamine, the next step of our signaling pathway is packaging. So we have to somehow package or store our dopamine so that it can be kept safely until we really need it. So let's look at how dopamine gets packaged. So now that we have our neuron, and inside our neuron we have our produced dopamine, we need to store it inside a vesicle, which is basically a storage compartment for our dopamine so that it can be released later on when it's really needed. So again, before we get into packaging, I just wanted to mention that I've already made a video dedicated entirely to packaging, and I think it'll help understand this video a lot better. So I'd really recommend watching that one first. I've linked it down in the description below. But let's go over it briefly. So you have your vesicle, right? And your vesicle has a couple of enzymes on it that help with this packaging process. So your first enzyme is called ATPase. And your other enzyme is called VMAT2. So here are your two enzymes, and they are on your vesicle. So now you have your dopamine, and basically the way that these enzymes work together to pump dopamine into your vesicle, this first enzyme, ATPase, what it does is it pumps protons into your vesicle. So you have protons floating outside your vesicle, and ATPase pumps these protons into your vesicle. So you have a buildup, accumulation of protons inside the vesicle, and accumulation of positive charge. And this accumulation of positive charge results in something called the proton motive force. The proton motive force. Basically, this proton motive force is a driving force that can be used to pump your dopamine into your vesicle. So basically now you have all these protons inside your vesicle, right? You have a bunch of positive charge thanks to your ATPase. Now your VMAT2 comes into play. What it does is it can exchange your positively charged cationic dopamine for your positively charged protons. It exchanges one positive thing for another. So it pumps out your protons and it pumps in your dopamine, effectively replacing your protons inside the vesicle with dopamine. And in doing so, it's able to get a bunch of dopamine into your vesicles where it can remain nice and safe and happy until it's really needed. And we have successfully packaged dopamine. Packaging is complete. So just to go over that again real quick, ATPase pumps in protons, creating a proton motive force. Then VMAT2 exchanges these positively charged protons for your positively charged dopamine, pumping it into your vesicles, and now your dopamine is stored inside your vesicle and ready for action when it's needed. So now that we've completed packaging, the next step of the pathway is release. Now that we've packaged dopamine inside our vesicle, we need to somehow release dopamine outside of this vesicle and into the synapse that follows after the neuron, so that it can go off and do things as needed. Now again, just like with packaging, I made a video already that's fully dedicated to release mechanisms, and I really recommend watching that one first. It'll make it a lot more clear, and I've attached that video in the description below. But let's look at how this works really briefly. So as you can see here, our dopamine is packaged inside our vesicle, inside our neuron. And here we have the terminal end of our neuron, which is the part at the end, and then afterwards we have this gap called the synapse. The synapse is the gap between a neuron and the adjacent cell, maybe another neuron or a gland or a muscle or something else, something that has receptors that neurotransmitters can go and act upon. But the gap in the middle is called the synapse. So in order for responses to happen, your dopamine needs to be released out into the synapse. We somehow need to get it out. So the way release works is via these 4 steps. They have to happen one after another. And as you can see, I've drawn little proteins here and here. So over here you have proteins that are attached to the vesicle's membrane, and these proteins are called V-snares or vamp proteins, attached to the membrane of your vesicle. And then over here, attached to the membrane of the terminal end of your neuron, you have these other types of proteins, which are called snap/T-snare proteins. And basically, these proteins interact with each other in order to allow for release. Let's take a look at how this works. So the first step of the release mechanism is translocation. And this one's pretty simple. As the name suggests, the vesicle makes its way over, or translocates, to the terminal membrane of the neuron. So it goes closer to the membrane, and it ends up somewhere, let's say it ends up here. So nice and close to the membrane. And this allows for the next step of the release mechanism. So after translocation, we have tethering. What happens in tethering is your vamp proteins and your snap proteins, they link up together, they attach. So they link up and they interact and they attach together. That's the second step of tethering. So then comes the next step of docking and priming. Now that your vamp and snap proteins have all tethered up together, basically docking and priming just means that more interactions and linkages happen between your vamp and snap proteins. Your vesicle likely gets pulled closer to your membrane as it prepares for the final step of fusion and exocytosis. And then with fusion and exocytosis, basically first what needs to happen is calcium ions need to enter the neuron. And then when calcium ions enter, this can trigger the final step of fusion and exocytosis. So what happens is your membrane basically fuses together with your cell membrane, and this results in your messenger molecules being released from your cell. So it's a little bit hard to show this with drawings, but basically what happens in your last step of fusion and exocytosis is your vesicle membrane fuses together with your neuron cell membrane, and when this happens, it releases all the dopamine inside your vesicle outside into your synapse. So this looks something a little bit like this. So your vesicle gets pulled really, really close to your cell membrane, and then they basically fuse together. And when they fuse together, it starts to look a little something like this. It starts to open up a little pathway, a little hole for dopamine inside the vesicle to be released out into your synapse. And now you have effectively released dopamine. So this last step is fusion and exocytosis. Fusion is the fusion of your vesicle and your cell membranes, and then exocytosis is the release of your messenger molecules, in this case dopamine, but this applies for lots of other messenger molecules. But the release of these messenger molecules out into your synapse. So we did it. We released the dopamine that was stored inside our vesicle out into the synapse. So again, I know the system can be a bit more complicated. If you're still struggling to understand it a bit, again, please watch that video I made all about release mechanisms. I think it'll help a lot. And also, be sure to check out this diagram, this picture of a diagram, which might make things a little bit more clear, especially that last step of fusion and exocytosis. I hope this helps a little bit. But that concludes the release step. We have released dopamine out into our synapse, and now it should be able to go and produce a response. So let's take a look at our next signaling step. So now that the dopamine has been released out into your synapse, this dopamine can now go and bind onto receptors on other cells. And it can go and bind onto these receptors and produce responses inside those cells. So dopamine can bind onto a variety of receptors on a variety of cells and produce a variety of responses. But one of the most common responses associated with dopamine is happiness. Dopamine regulates mood. It makes you feel happy. It makes you feel pleasure. That's one of the main things dopamine is associated with. Another thing dopamine is associated with is something called the sympathetic nervous system. And this is something that we're going to look at a lot more in a future episode. Basically, your autonomic or automatic nervous system is split into two halves. You have your sympathetic and parasympathetic nervous system. Your sympathetic system, you probably heard of this term before. It controls your body's fight or flight response. So when you're in a dangerous situation, when you either need to fight back or run away, that's your sympathetic system, and your body goes on high alert mode, high heart rates, high breathing, all that. And then on the other hand, your parasympathetic system is when you're not facing any danger. This is your rest and digest. Your body is at rest, your digestive system is working. This is your normal states. So dopamine is heavily involved in your sympathetic nervous system. Its responses are responsible for this system. And in the future video where we look more in depth about the sympathetic and parasympathetic nervous system, we're going to look at these responses more in depth. So stay tuned for that video. But for now, just know that dopamine is involved in the sympathetic nervous system, and it's also one of its main responses is mood regulation and happiness or pleasure. And with that, we've gone over another step of the signaling pathway. We've looked at dopamine's responses. But as we know, responses, if they go on indefinitely, this can be very dangerous. So we need to terminate these responses to prevent them from going out of control. Now, again, I know I've said this again and again, but I've already made a video all about common termination mechanisms, and I think that would really, really help understand the termination mechanisms specific to dopamine. So again, I'd really recommend watching that one first. But yeah, let's get into it. Alright, so the first main termination mechanism is uptake. So now that you have dopamine in your synapse, one way you can get terminated is by getting reuptaken into different cells and get stored for there so it can be used later on. So your dopamine, it can be reuptaken into the presynaptic neuron which just released it, and then it can get stored inside there for later use. Or it can get uptaken into other non-neuronal, so just other types of cells, and it can get stored there for later use. But the general idea is the same. It gets uptaken into cells and stored there until it's needed. And that's uptake. Pretty simple. The next termination mechanism is enzyme degradation or enzyme conversion. So enzyme degradation, as the name suggests, basically enzymes of the body can degrade or inactivate messenger molecules, and this applies for dopamine. So two examples of messenger molecules that can terminate dopamine are monoamine oxidase, and catechol-O-methyltransferase. So monoamine oxidase is made in the mitochondria of organs like your liver and the epithelium of your intestines. And it acts on many messenger molecules such as dopamine, but also serotonin and noradrenaline. And what monoamine oxidase does is it oxidizes dopamine. It combines it with oxygen. And what's interesting is that monoamine oxidase, remember, it terminates dopamine response, right? Well, some antidepressant drugs can actually inhibit monoamine oxidase. So antidepressant drugs can stop monoamine oxidase from terminating dopamine. So basically, by stopping monoamine oxidase, they stop the termination of dopamine and they allow for increased dopamine response. And that makes sense because we mentioned previously that dopamine is linked to feelings of happiness and pleasure and mood regulation. And antidepressant drugs, they by increasing, by allowing for more dopamine signaling, they increase your mood, which helps them lower depression. So that's kind of interesting. They work by inhibiting your termination mechanism of monoamine oxidase. So monoamine oxidase, that's one of the messenger molecules that can terminate dopamine. But another one is catechol-O-methyltransferase. What catechol-O-methyltransferase does is it transfers a methyl group onto your dopamine. And actually, as the name suggests, catechol, it can do this to a variety of different catecholamines. It can act on a bunch of different catecholamines, add methyl groups to them, and terminate them. So these are your two common termination mechanisms for dopamine. One of them oxidizes it, and one of them adds a methyl group, but they both effectively inactivate and terminate dopamine signaling, keeping dopamine responses under control. Now, let's look at our third mechanism of receptor desensitization. So the third termination mechanism, which is called receptor desensitization, it's a little bit different than our first two mechanisms. Whereas uptake of our messenger molecule and degradation of our messenger molecule, they both act on our messenger molecule, in this case, dopamine. But receptor desensitization, it doesn't do anything to the dopamine, it instead acts on the receptors, as the name suggests, receptor desensitization. It acts on the receptors of the postsynaptic cell. So basically, when dopamine is released from the presynaptic cell into the synapse, which is a gap between your two cells, then dopamine can go over and bind onto these receptors on your postsynaptic cell. And when it does so, it can initiate a response inside your postsynaptic cell. So if we somehow deactivate or get rid of or do something to these receptors, then dopamine can no longer bind onto them because there's no receptors to bind to. And then if it can't bind onto the receptors, it can't produce a response. And that's how receptor desensitization works. It acts on these receptors to prevent dopamine binding and prevent response. And it does this to keep responses under control. Now, like I've said before, I already made a video all about termination mechanisms, and in this video we read really in depth to receptor desensitization, because this is actually a pretty complicated process with a lot of steps, so we won't be able to get through it in detail in this video. So if you want to learn more about receptor desensitization, be sure to check out that video, but let's go over it somewhat briefly in this video. So basically, right, in your synapse, but basically before your postsynaptic cell, you have dopamine, right? Now, let's say you have an excess of dopamine. Let's say there's a bunch of dopamine. I'm going to write D for dopamine. Let's say there's a lot of dopamine, right? All of those dopamines, they're going to want to come and bind onto your receptors. So if you have a lot of dopamine, then you're going to come and bind onto a lot of receptors, and they're going to produce lots and lots of response inside your cell. An excessive amount of messenger molecules and binding onto receptors can produce an excess response. And the whole point of termination mechanisms is to prevent excess response. We don't want response to get out of control. So what receptor desensitization does is it basically inactivates these receptors. And by inactivating these receptors, it prevents dopamine from binding onto them. It doesn't completely prevent it, but it lowers the amount of dopamine that can bind onto receptors by taking away some of these receptors. Now you only have, let's say, 2 receptors instead of 4. So not as much dopamine can bind and not as much response can be produced. Only, let's say, half the response can be produced. So just before we get into it, let me just reiterate again. When you have an excess of messenger molecules like dopamine, they can excessively bind onto your receptors. So by desensitizing or inactivating some of these receptors, let me just erase them, you now have less receptors for your dopamine to bind onto, and therefore you can produce less response. So how do these receptors get inactivated? Well, they actually get transferred. You see how here they're like on the outside of the cell? What happens is they're actually pulled inside the cell, and they stay inside the cell in these little compartments, kind of like vesicles, but they're called endosomes. So we've transferred our receptors from outside the cell to inside the cell. And now now that they're on the inside of the cell, dopamine cannot enter the cell to bind onto them. So that means that, now we're only limited to 2 receptors instead of 4. And basically, these 2 receptors that we now have inside the cell, they can stay here for a while, but then eventually they can get resensitized. They can return back up to the surface, and they can reattach to the cell membrane, and now they are ready for action again. So dopamine can come and bind onto them and produce response. But let's say, again, we have too much dopamine, too much response. Well, then desensitization will happen again. Maybe this time, these receptors will get desensitized. They'll be pulled into the cell in their endosomes. So it's like a cycle. We have a bunch of receptors outside. If they have excessive signaling, then we pull a couple inside because it gets desensitized. Then we resensitize it and the receptors go back up. But again, if there's too much signaling, then they'll have to be brought back inside again. So it's like a cycle of desensitization and resensitization. So in case it wasn't that clear, I've drawn a little diagram here. Hopefully this can help. So as you can see, you have a lot of dopamine binding onto your receptors. And because we have excess response, a couple of your receptors, these 2, are getting internalized. They're getting pulled inside the cell, inside these endosomes. And then after some time, they can get resensitized and return back up to the surface. And now they are active receptors again, and dopamine can come and bind onto them. But eventually, if there's too much response and the cycle repeats, more receptors can be pulled inside the cell. And it's basically a delicate balance of desensitization and resensitization that overall keeps responses under control. Now, one important thing to note is that if this goes on for a long amount of time, like let's say we have a lot of dopamine, a lot of signaling, an excessive amount of response for an extended period of time, then actually, instead of resensitization, instead of the receptors being returned back up to the surface, something else can happen. These receptors can instead to go over to something called a lysosome, another organelle with digestive enzymes. And what happens is the receptors go over to lysosomes and they're actually broken down. So they permanently get rid of these receptors. So basically you have a temporary termination mechanism and a permanent termination mechanism. The first one we looked at, your dopamine receptors are brought inside the cell, and then after some time they get resensitized. So resensitization, I don't even know if I spelled that right, but this is a temporary termination mechanism because it's only for some time. After a while, those receptors can be returned back up to the surface and all is good. But in extreme cases where we have so much signaling and this just isn't enough, we need a more permanent solution, then we can instead send those receptors to lysosomes to get broken down. And that is what we call down regulation. And this is a more permanent termination mechanism, because when we send these receptors over to our lysosome and have them permanently broken down, well, now your receptor is gone. Your receptor is just broken. It cannot be returned back up to the surface. The total amount of receptors you have has decreased since you've just broken some of them apart. So it's a much more permanent termination mechanisms for extreme cases of excessive signaling. So in conclusion, receptors can be desensitized, brought into the cell, and depending on the situation, they can either get resensitized or downregulated. But that covers our third termination mechanism. And with that, we have concluded the last step of the signaling pathway of termination. And that basically concludes the dopamine signaling pathway. We saw how dopamine gets produced, how you go from phenylalanine to tyrosine to L-DOPA, and then finally to dopamine. We saw how dopamine gets packaged into vesicles with the help of enzymes like VMATs and ATPase. And then we looked at how proteins like SNAP proteins and VAM proteins help dopamine get released into your synapse, and then how dopamine can bind onto receptors to produce a response, such as controlling your mood, your happiness, reward mechanisms, and how dopamine can also be involved in your sympathetic nervous system. And then we finally looked at how dopamine gets terminated, either by enzymes or by getting reuptaken into cells, or by having your receptors get desensitized so that dopamine can have a harder time binding onto them to produce a response. But that concludes this signaling pathway. Now remember we mentioned how dopamine is a catecholamine, meaning it is an amine that has this catechol structure. Well, it turns out that there's a few different types of catecholamines, and in the next video, we're going to explore the catecholamines norepinephrine and adrenaline. So these are two different types of catecholamines that we're going to explore in the next video. So this was basically catecholamines part 1. Next video will be catecholamines part 2. But until then, thank you guys so much for watching. I really appreciate it. If there's any suggestions or tips or feedback you'd like to give, anything you want me to go over again, maybe explain better, please drop a comment below. I'm open to any feedback and I'd really appreciate it. But yeah, until then, thanks again for watching. I hope you enjoyed the video and I'll see you guys next time. Take care and have a great day.